Hello everyone, my name is Nayanika and I'm doing a PhD at University of Vic, Central University of Catalonia. The topic of my PhD is bringing together molecular simulations and molecular evolution, and this is one of the projects of my PhD. So in this, what I study is a class of proteins which is a selenocysteine class of proteins and a cysteine class of proteins, GPX6. So GPX is glutathione peroxidase, and we specially study GPX6 because it has a cysteine in the rodents and selenocysteine in humans. It is one of the isoforms of GPX. What does GPX do? So it actually maintains the reactive oxygen stress of the body. So this is the balance. And the mechanism of GPX has been studied by Murukuma like 20 years ago, and this is the general catalytic cycle of it. While when we go into the details of the mechanism, we know there is a proton transfer that happens in the first step and in going from selenol to selenolate and selenolate to selenic acid. While this can also happen in a concerted way. So in this concerted way, we do not have the steps. We just have one step which goes in formation from selenol to selenic acid. While when we look at the conformational differences between the different isoforms, there is not much difference which is seen. All of them are folded in such a manner that the active site happens to be in the same position. While when we also see that the glutathione disulfide, which is the byproduct, when it binds to the active site, it is also located in the same way. So this tells us that in conformational way, these are all when we look at the conformational side of it, they are all similar to each other. They are homologs. What we actually want to understand is the evolutionary shift that has happened from cysteine to selenocysteine. So we have a central question which says that GPX6 appears to lose its peroxidase activity when the selenium is replaced by cysteine in the active site. So does this happen only because of this one mutation, or is it because of a lot of mutation, which generally means epistasis? So we want to understand the epistasis of this accumulation of the mutation. In doing so, the methodology that we use is empirical valence bond simulations, which is done with the help of a program called Q in calculating the free energy perturbation of all of this to understand the electrostatics of the system and the overall conformational changes that happens in the system. So in selecting the variants, we had a strategy in selecting them, since we want to understand what happens when a mouse is completely transformed into a human and a human is completely transformed into a mouse with cysteine and selenocysteine. This is the approach that we used. So we did an MSA, that is multiple sequence alignment, and when we did that, we found like 46 different places where the amino acid were different in mouse and human. And when that happened, this table tells us that all these residues are different in both these enzymes. So we slowly changed one residue at a time in mouse and in human, along with the essential 49th residue, which is the cysteine or the selenocysteine, to understand what is the different electrostatic effect that can occur in them, as well as if they show a drastic conformational change. And this was also done in a certain category. Now that category was the distance based category, which we can see in this plot. So all of these positions are those mutations that we have done in these two sequences, and the red line shows that there is selenium in that position and selenium or cysteine at that position, which is 49, and we calculated the C alpha distance from all of them to understand which mutation is located in which part of the graph. In that way, we categorized them as 0 to 10 angstrom, 10 to 15, 15 to 20, 20 to 25, etc. And based on that, this is the first result that we have, and this is going from 0 to 10 angstrom, which is very close to the active site. Also, this is for the first part of the reaction 1, which is going from selenol to selenolate, where it loses the proton. What we see here is overall the table, and this bar graph shows the mouse and the human delta G star and delta G0, which is the free energies and the reaction free energy. So when we see at the activation plots, these are what we actually expected to happen, because we had a hypothesis that the selenium reacts faster than cysteine, and which has been proven here, because yes, the selenium has a lower activation barrier than the cysteine. So the human selenium has the lowest activation barrier, and the mouse cysteine has a very high barrier. And when we compare all others with the mouse cysteine, we see that all the barriers are lesser, which are green in color, while here we see that all of them are lesser than all of them are more than the human selenium. So this tells us that the selenium is is having the least barrier, and it is reacting really fast. So this was one of the conclusions that we had from these, and the other conclusion which are still ongoing and which I think would be more better understood when we calculate the electrostatics of the other mutations as well. The difference that the mutation makes in the barrier. So here we see that this, when we change from cysteine to arginine or from serine to ala, there is a little difference, and there is a loss of hydrogen bond as well. So these things are happening, and that's why we see that there is a change in the barrier of when the mutations are added. So that was another conclusion, but we still need to specifically understand this more. While when we check at the conformational level, there is not much difference seen in them. All of them have the same structural features, structural alignment. The stability is not much different, though there are some peaks at certain points, but they are not seen in the active site. Similarly, when we did another approach, which was running a longer simulation for almost 400 nanoseconds for the four systems, that is human and mouse cysteine, cyanocysteine, along with the glutathione disulfide, which is the byproduct. We did not see significant differences also in the RMSF plots, which you can see here. And this also tells us that the overall fluctuations in the dimensions do not change, because, as I said before, that the conformation is quite similar. They are homologs of each other, so it tends to be aligning to that feature as well. So the next part of this is running more mutations to understand the metrics better, the electrostatics of the system, and doing the step 2 as well, because the step 1 would be like this portion, but then the step 2 would be the final formation of selenic acid, which is the important part as well. And then running all these simulations in the ancestor, which has been reconstructed, and that would help us to understand how from the ancestor to the modern mouse and human, how is the evolutionary pathway linked, and that will also help us to understand if there is loss or if there is a gain of the peroxidase activity, which will be the final conclusion of understanding what is the function of GPX. Thank you for hearing me, and if you have any further questions, I'm open to all of them. Thank you so much.